Thank you. Thanks a lot for the introduction. And let me start talking about food safety, because even though the US has a very safe food system, it is still problematic. There's 1 in 6 Americans getting sick every year. There are thousands of related hospitalizations and some deaths. When you take into account productivity losses, the healthcare cost, it actually costs $70 billion a year to deal with food safety. So it's still an important problem. And what is interesting is that things are not getting better, even though entities like USDA or FDA are trying really hard to bring these numbers down, they are not improving, or at least not as much as they want. And they have identified 2 important reasons for that. First is that you have a biological enemy. So when you start to get to know one of these organisms, then another one may evolve, mutate, and they become dangerous. That's what happened in 2011 in Germany, and in that outbreak, 60 people died. Another reason is that right now we have a very complex food system. When you have this plot here is showing the value of agricultural imports into the US. It actually has doubled in the last 10 years. And when you have food coming from Mexico, peanuts from Vietnam, and many times all of them are putting the same product, then it's really hard to track those pathogens, or just make sure that the quality is good. Now, there are different approaches to tackle food safety. One of them is education. So for example, don't get your burgers medium rare. You want to get them well done, because in the pink part there might be E. coli. Or also, if you like to cook rice and then leave it overnight, put it in the fridge, because otherwise you just have a perfect media for bacteria to grow. So, those education tips are important, and it's a way to tackle this issue. But another one is to create new sensors that let us detect the presence of pathogens in food. Now, my group has been working in point-of-care devices. So point-of-care devices are these small, portable, inexpensive, and simple diagnostic tools. Those are a few examples of how they look like. The idea is that they're tiny, and the user can just go and take samples from the patient and analyze it. And what they bring is they have a fast turnaround time. You can get diagnostics very quickly. They also reduce the cost to get data, and also overall they improve patient care. Now, those characteristics of speed and reduced cost and portability, of course, are also interesting for the food safety industry. They also want to have those same abilities in these sensors. So we started to think, well, how can we use that knowledge that we have in point-of-care devices and translate it for food safety? So this is our solution. We're doing electrical detection of a DNA amplification reaction that is known as LAMP. And in order to do that, we use semiconductor sensors that are known as ISFETs. So then here in the picture you have the ISFET sensor, which is the brown part here. Then there are microchambers, and in each one of those chambers we're going to be conducting one of these reactions, one of the LAMP amplification reactions, and we get an electrical signal. And the overall goal of this is that with this kind of device, you could do pathogen detection with a miniaturized setup, very inexpensive, by the way, because this is just transistors like the ones you use in your cell phone, in a portable setup. So that's the overall goal. And here in the cartoons, I'm showing the different steps that you have. First, you need to dehydrate primers. Primers are reagents that bring specificity to the reaction. Then you inject the sample that you want to analyze. And if the template finds a matching primer, if the pathogen that you're looking for is present in that sample, then you're going to get an electric signal in the form of a pH change. So that is a very quick way to explain what I'm doing, but because this is a tutorial talk, then I want to explain with a little bit more detail the concepts and all the acronyms that I was throwing. So let's start with LAMP. So LAMP, for those of you that know about molecular diagnostics, LAMP is the isothermal equivalent of PCR. So if you don't know what PCR or LAMP is, the idea is that you're going to multiply DNA exponentially. You create a lot of DNA, but that only happens if a target sequence is present in the sample. If a target sequence is not present in the sample, then you don't have any amplification, and therefore you can correlate that with the presence or not of a pathogen, because the pathogens have a very specific sequence. Now, that specificity, that reaction or no reaction, is dictated by the primer set that you have, and that is what I was mentioning before. So overall, that's how the amplification reactions work. You only amplify DNA again if the pathogen is present. Now, LAMP, going back to LAMP, LAMP is very similar to PCR, but it has certain advantages, especially for point-of-care devices. First is isothermal. While PCR, you need to thermocycle. Here, you just hold it at 1 temperature, and that's convenient. It's also sensitive and specific. It has a high amplification efficiency, and it's also robust. It's a little bit more tolerant to inhibitors, so it has advantages over PCR. Now, the second acronym was ISFET, and that stands for ion-sensitive field effect transistors. So if you're familiar with MOSFETs, which are these ones, and how we make memories and processors and all those other things. So you have here the MOSFET, and here, just to compare it, is the ISFET. Now, they are very similar structures. The main difference is that here, instead of a gate connection, you have replaced that with a fluid and a reference electrode. Now, under this configuration, the flat band voltage of the transistor, which is related to the threshold voltage and therefore the drain current that you're measuring, becomes a function of something that is called the surface potential, which is the potential that you have between the electrolyte and the dielectric of the transistor. And that surface potential is also a function of molecules that get close to the gate. So a lot of people have been using these ISFET transistors to detect charged molecules in a fluid. And also you can use it to detect ions. Ions are going to form a double layer, what is called the Helmholtz double layer, and that changes the potential of the transistor. Therefore, it changes the threshold voltage and then the current, and you can detect electrically ions using ISFETs. And it's a very elegant way to do it because, of course, we're leveraging what the semiconductor industry has done for the last 50 years, and now we can make millions of these devices, as I will show you, in a very tiny area, getting a lot of very important sensing power. Now, those two concepts, the ISFET and LAMP, get together in the following way. It turns out that that amplification reaction is going to give you a pH signal. And because you can sense pH signal with the ISFET, therefore you can detect the reaction using electrical semiconductor transistors. So this is a cartoon to explain how that happens. The idea is that when you're replicating DNA, both with LAMP and PCR, you have 3 nucleotides in the solution. There's a polymerase that is going to incorporate a nucleotide into the elongating strand. So the polymerase adds the different base. This is my T and my C. It's just a cartoon, but what is important here is that you have a couple of byproducts. You're kicking out a pyrophosphate. This is called a pyrophosphate, and also a hydrogen ion. Now, because you're increasing the quantity of hydrogen ions in solution, you're changing the pH by definition. So then, as the reaction progresses and as you're multiplying DNA, then the pH of the solution is changing. And by the way, this is the same concept that IonTorrent, which is a sequencing company, is using in their sequencers. That's the idea, that you can detect amplification reactions based on pH changes. So hopefully now this is a little bit more clear. The idea here is that you prepare the chip with a primer. The primer, again, brings specificity to the reaction and only triggers amplification if the pathogen is present. Then you bring your sample, and if the primer that you put here is compatible or recognizes the DNA strand of the pathogen that you're looking for, then there's going to be amplification or multiplication of DNA. And that multiplication is related with a pH change that then you can electrically detect with your ISFET in a very miniaturized platform like the one I was showing before. So those are the concepts. Now, going back to the tutorial talk, this is what you will need to perform one of these reactions. These are the 5 elements that you will need to use to detect pathogens in a food sample. So, of course, you have a sensor, and this is where we have put most of our research efforts. We have a chip that has 1 million of these ISFETs in a 7 by 7 millimeter area. That's going to become important later, as I will show, because you need redundancy in your measurements to get a good signal-to-noise ratio. But overall, you have a million different of these devices. Each one of them is tracking pH changes, and therefore they can potentially detect the reaction. So here I'm just showing how, depending on the pH, is gonna change the current of the transistor, and there's a related distribution, because remember, there's a million of these devices. Then the second part is the reaction mix. So in the reaction mix, you have a polymerase, the free nucleotides, the sample that you want to analyze, and that's going to be inside the reaction chambers that I was showing in the previous pictures. Then you need a microinjector, and although we're working to switch to microfluidics to make this more portable, so far what we're doing is to inject samples inside the chip using a microinjector. So this is basically a very tiny pipette, and you can just put volumes inside the chip. You need a heater also because the reaction happens on 65 degrees Celsius, so you need to heat up the chip in order to trigger the reaction. And finally, you need some circuitry to put the voltages on the transistor that you want, and also record the currents that you get out of the reactions. So those are the elements that you need. Now, if you have these things, then the idea is that first you dehydrate primers using the microinjector. You can, in each one of those micro chambers, you can put a specific primer. In theory, you can have a primer per chamber, so you have multiplex assays. What we do so far is to use many of them to target the same pathogen, because that way you can have replicates and get more robust data. So that will be the first step. Then you need to put mineral oil. The droplets that we're putting are 250 nanoliters, so they can evaporate very quickly if there's no barrier for evaporation. So then you put mineral oil, and then you inject the rest of the lamp mix, including the sample that you want to analyze. And that will be what we call the setup for the assay. Now, once you have all this prepared, then you can run a reaction. And then the idea here is that in this experiment, I divided the chip in 3 different portions. In one of them, I have primers for Listeria, in another one for Salmonella and E. coli. So that's the preparation, injecting the primers. Now, in the second step that I was showing in the previous slide, I was injecting E. coli, or lamp mix that includes E. coli. So we're only expecting DNA amplification from the wells that have the primer, which are the 2 columns in your right. Then you take fluorescence images and an electrical image. The standard procedure to do to monitor DNA amplification reactions is using fluorescent labels. So we're still doing fluorescent labels just to confirm our assays. But then we're also detecting or measuring the current of each one of the transistors for a comparative analysis. Then what you do is you heat up the chip. You heat it up at 65 degrees Celsius. And you get amplification only in the chambers where you are having both the template and the primer that are adequate for that reaction. There's also a current differential signal. However, that differential signal is very small. We're talking about 2% or 5%. So that's the reason having many transistors becomes important, because that way you can filter data and do robust statistics to get an accurate perception of what's going on with the reaction. So once you finish your reaction, you collect your data. The data looks like current populations. You have different current populations in each one of the chambers. There's about 10,000 transistors monitoring each one of the reactions, so there's a distribution related to that reaction. Then what we do is we try to get the best signal-to-noise ratio that we can out of our data. And in order to do that, we filter data. And there's 2 steps to filtering the data. First, you can discard sensors that don't have the adequate performance. For example, sensors that are too noisy, that are variating too much, you can disregard those, because those are not representative of what's happening. Then the second tier for filtering is use statistical filters. So there are a couple tests. One of them is called the Grubbs test, and then the other one is ESD, and that lets you figure out abnormal points that fall away from your normal distribution. So this is an example where this portion was not part of the normal distribution, and then the test will discard it. And then finally, after you do all these things, then you have a signal between the chambers where there was no amplification and the chamber where there was some amplification, meaning that that pathogen that you were targeting with the primer that you dried, it's present in that sample. So these are the different distributions. So E. coli had a larger shift, a greater increase in current than the other chambers. And then when you compare it with statistical tests like p-values and ANOVAs and things like that, then you get significant difference between the different chambers. And that will let you know, well, you have E. coli in that sample that you were putting in the chip. So that is overall the idea that we're playing with. We want to create these point-of-care devices for food safety. You can do LAMP reactions to detect pathogens and then detect those reactions using ISFETs. The idea that we have for multiplexing can be done with primer dehydration. So when you have multiple primers, you can be targeting multiple pathogens, which is important for the food industry because they want to have a comprehensive screening assay. And then ideally, this will be used by people like USDA or FDA to have a better control over the food safety and reduce those numbers that I was mentioning before. And yeah, thank you to the funding agencies and my group. Thanks a lot.